Okay, well, hi. I hope everyone's doing well. As Dr. Wilkes said, my name is Iman Haddad, and my thesis is on the effects of anthropogenic activity on gray fox spatiotemporal activity and dietary behavior. And actually, my whole thesis was learning to pronounce Erosion Scenario Argentius. So that's it. You can leave now. But I know that was a mouthful, so let me give you guys an introduction and break it down. So first, the effects of urbanization on wildlife. So urbanization is something that is increasing rapidly in Texas. Our cities are really growing quite rapidly, and our population is getting denser. And Austin is a city that has been heavily impacted by this trend. And as urbanization increases, wildlife are going to be forced to either adapt their behaviors or to essentially leave. And so what scientists have found is that 93% of observed behaviors across urban wildlife studies have had significant differences compared to their wild counterparts. So we know that urbanization is severely impacting our wildlife. One of these behavior changes that you might see is that activity times in crepuscular species, which means species that are more active around dawn and dusk, may shift to be more nocturnal in order to avoid human conflict in urban areas. Another thing that's really important to note is that one urban space that a lot of wildlife may interact with is a college or university campus. And campuses across the US are generally trending towards a perspective that aims to attract wildlife. So thinking about planting native plants and plants that wildlife can eat, as well as making landscaping decisions and installing features that are usable for wildlife. Now, gray foxes in urban environments specifically. So gray foxes are a mesopredator omnivore. So they eat both plants and animals, and they primarily eat small mammals as well as birds and other small prey. And they're considered urban neutral, which means that they, more than a lot of other species, are able to adapt and even thrive in urban environments. And they're one that are more likely to be seen in an urban environment. If you live on campus, you might have seen them here, but they're all across Austin. Research suggests that they are a hardwood-dwelling species. They climb trees, as you can see in that really cute picture that Dr. Ledesma took. But more recent studies have found that in urban environments, some foxes are actually showing preferences towards open fields. So they're actively changing their temporal ecology and what they're using spaces for. Or sorry, not their temporal, their spatial ecology. And also, they've been noted, as I said, with crepuscular species, they are active later at night than their wild counterparts in areas that have higher levels of human presence. Lastly, there has been a noted decrease in avoidance between wildlife competitors in order to maximize avoidance of humans. And this is important when thinking about foxes and one of their other mesopredator competitors, coyotes, both of which can exist in Austin. So the questions that sort of drove this research: one, how do anthropogenic landscapes influence gray fox spatial use? Two, does anthropogenic presence affect gray fox temporal activity? And three, what are foxes eating on campus, and how does it differ from what they eat in natural environments? And the predictions that we made based off of this, one, the more heavily human-modified areas of campus will see fewer instances of fox presence compared to natural areas. Two, sightings of foxes in human-modified areas will occur on average later at night compared to natural areas. And three, campus foxes will exhibit an altered dietary composition from their wild counterparts. For the methodology, so I'll give you a little bit of site background for those of you who aren't from St. Ed's, or even if you are, maybe you'll learn something new. So St. Edward's University is located near downtown Austin, Texas. It's also near the I-35 highway, and our surrounding neighborhoods are South Congress and South Lamar, which are both highly populated, very high traffic neighborhoods. And so all of this places St. Edward's within a very highly urbanized context, But in the midst of that urban context, so St. Edward's has 60 acres of land, and this land includes a protected forest area called the East Woods. And we are also directly across the street from Bloom Creek Nature Preserve. And on top of all of this, St. Edward's has been known to be a champion of providing habitat for wildlife, reducing pesticide use, planting native plants. And so all of these make St. Edward's somewhat of a possible wildlife haven within an urban environment. So for site selection, we chose 6 locations across the St. Edward's campus, and these were designated as either human-modified or natural. The human-modified sites were characterized by lower vegetational coverage overall, and were also known to get heavy foot traffic from students, faculty, and staff, typically because they had more infrastructure like sidewalks or were an academic building or dining hall. And then the natural sites had higher overall vegetational coverage, and they received lower levels of human foot traffic, often being on the edges of campus. So these are the sites. The human-modified sites were John Brooks Williams Natural Science Building North, and then the Ragsdale Lawn, and then a trail by the soccer field. And the natural sites are a known den location behind Equity Hall, the campus retention pond, and the campus sustainability garden and food forest. So to measure their spatiotemporal activity, which again refers to the spaces that they're using and their time of activity, we had traps set to capture motion at each site from 5 p.m. to 9 a.m., so that would be their expected time of activity. And what we measured was the average number of sightings per trapping period, so that 5 p.m. to 9 a.m., and the range of the time of appearance of foxes per camera. And for dietary analysis, so we, throughout the spring semester, collected fox scat, which you can see in that picture there, really beautiful, from across campus all throughout the semester. And for each piece of scat, the location was then logged to Q fields for QGIS, and they were numbered. And we would take a photo with a color scale. Color scale's not in that one, sorry guys. And then we would freeze the scat and place it in an autoclave, and then we would break the scat that was then sanitized down under a microscope and essentially pick out any identifiable food items for analysis. And so for statistical analyses, I did Welch's t-test to compare the human-modified sites to the natural sites for the number of sightings per trap period, as well as the activity times. And those analyses were based on data collected throughout the fall semester of 2025, so last semester, October through November. And then fecal samples, the current project isn't doing any statistical analysis. We're just looking at what they're eating and kind of building a baseline. And all analyses were conducted in our studio. So for the results, first of all, there's a really cute fox. That's by JBWN. That's the most important result, of course. So for the number of foxes at each side, I'll point out to you those yellow bars represent the natural sites, and the blue ones are the human-modified sites. And you can see that in general, the natural sites had just throughout the semester more instances of fox presence. And then when comparing human-modified to natural and taking those averages by camera, you can see that there's a significant difference, that those natural sites, again in yellow, had a significantly higher average fox instance per trap night throughout that semester. And then I ran it again because I had technical issues with the camera, so I kicked it out and it became even more pronounced. So you can see, even with those technical issues, there's still that clear difference. And then when thinking about their temporal activity, so what this plot is essentially showing, you can see, I'm gonna point your attention mainly to those dots on kind of the bottom parts of the violin. And what you're seeing there is that even though their start time of activity was typically the same, the foxes were actually lingering at those natural sites longer into the morning, up until that sort of 9 a.m. end point of when the cameras were set to capture motion, whereas in the human modified areas, they were stopping around 6 or 7 a.m. and there weren't any later morning sightings. And so what that's really showing is you can kind of see that that halt sort of correlates with when people start showing up for classes and faculty start coming into their offices. And so we're seeing a sort of difference in their temporal use of spaces as well. And then this is just a really cool graph to show you that we got a lot on campus, including people in all of these cameras. And you can see there were foxes at every single location. And so they are using pretty much the whole of campus. But you can also see that in the sites where, the pond, there were a lot of people that were captured, but it's also the site that had the most foxes. So that's something to think about if they're being accustomed to human presence or if they're just altering their timeline to avoid people. But then also the next highest one for fox presence was behind Equity Hall, and we had no people on the cameras during that sampling period. So that's something really interesting to think about. And for the scat, so these are some of the interesting items that we found. One is bird feathers, which means that they are hunting on campus. They're catching birds. Unfortunate for the bird, great for the fox. But on a less happy note, they can also eat cookie wrappers. And we did pick out, it was a Keebler fudge cookie wrapper, if anyone's wondering. And in that same piece of scat, also found some synthetic fibers. And so this is showing that foxes are, through our campus, getting access to potentially harmful food items. On a more positive note, we also found some seeds and other plant materials. And so that's showing that they are still eating the plants on or near campus, and they're able to utilize what we're planting. And we also found an insect exoskeleton. You can see it's that one on the left that's next to a seed. So those were some of the things that we found so far. So now, what did we learn? The key findings are, one, as I mentioned, the natural locations exhibited significantly more instances of fox presence than the human modified sites, which goes with our first prediction. It supports that prediction. Also, foxes were observed in natural sites later into the morning compared to human-modified sites, so they are adjusting temporal use because of anthropogenic activity. And foxes on campus have access to potentially harmful food items, such as that wrapper that they consumed. And what that's really telling us is that we need to think about how we're managing our trash and, what wildlife are potentially harmed by how our lifestyles are. Some limitations with this study. I had some technical issues with the camera, so the data set is smaller than I would have ideally liked. I would have loved to have data from this semester as well and from winter. And also the data analysis and collection is really time intensive, and so our scope had to be rather narrow, focusing on campus, but ideally a future study, more hands, more time, would be able to compare campus to a wildlife preserve to really get an idea of an urban versus natural setting. Some future directions, if someone wanted to pick up this project and continue. One, a continuation of this project would be able to provide analysis for all seasons on campus, and that way we would be able to represent both a semester when people are here and see if there are any seasonal differences when people are not present, such as winter and summer breaks. Future SCAT analysis should also aim to identify down to species what we're picking out, so not just the seeds, but also knowing what plant that seed came from, what bird those feathers came from, and also calculate the % frequency of occurrence of each food item to see if the composition is altered. And looking forward, this research can really give us a better understanding of how our lifestyles are impacting wildlife on campus and how we can really manage our campus to be better for wildlife. Another potential future project, as I mentioned earlier, foxes and coyotes are competitors. And I found a study that talked about how gray foxes and coyotes, they're both mesopredators and they're competing for the same resources. And foxes often climb trees in order to avoid coyotes. Gray foxes are more likely to inhabit a high human density area than coyotes are, but that doesn't mean that coyotes won't be present. And so this study found that fox activity did shift to be more nocturnal when coyotes were present, regardless of the amount of forest cover. But in areas with less forest cover where these foxes couldn't escape, these results were more pronounced. And this could be really relevant to the camera trapping and for someone to study that on St. Edward's because we've had a coyote before, and that's in front of Ragsdale, so right in the middle of campus. And so thinking about how our campus and our landscaping are going to affect those interactions and that competition will be really important. And with that, there are my resources. Thank you all for coming. I'd really like to thank my... And I'd love to thank everyone who helped me along this way. My advisor, Dr. Ledesma, and also Dr. Consilio and Dr. Johnson are here, and they gave feedback, and Dr. Johnson is helping me make maps in GIS for this project, so it's really cool. I really appreciate everyone who helped.
That was fascinating, and for the first time I think I've seen a giant picture of poop in a gold star. Any questions for Iman?
Yeah, Dr. Consilio.
So the pond data is interesting because your cameras are set just it was from 5 p.m. to 9 a.m.
Mm-hmm.
So people are walking around that area at night? Or
Yes.
Okay.
Yeah, so I was only taking any data that was from those periods.
So did you get a chance to look yet at whether there is any difference in the time when human activity is the greatest around the pond versus fox activity?
I haven't been able to categorize it statistically. What I can say was that when I was looking through and doing all of the ID, there was actually a pretty big gap between when there would be a human sighting and when the next picture of an animal sighting would be. So as I was combing through, just anecdotally, there was a gap. So there is that avoidance.
Yeah, that's cool though.
Bill?
Do you know if the scat that had synthetic fibers and that wrapper, do you know if that was found or collected at a likely human modified area?
Yeah, so that one was actually collected, if you're familiar with the soccer field and how there's a hill that kind of goes up for it, and it's kind of near that corner that by the road actually, and then loops around to where the operations building is. So that's where it was, but what I will say is that where the scat is left might not always be where the food item was consumed. So there's not really a way to know where it got it.
Well, I was just sorry, just to follow up, the range, do you know the average range of an urban fox versus would some of these individuals be using the whole campus?
I think they are using pretty much the whole campus. It's probably a smaller population of foxes using the whole area, and not necessarily a ton of different foxes, because they're rather territorial actually, and they defend that area.
Do you know if anybody would continue your research?
I don't know yet. I really hope that another student does and wants to pick it up, because that would be really cool and interesting. So hopefully someone is fascinated and wants to.
It would be nice to have students do a campaign so that the fox is in the
Yeah.
Yes, I get to call up, yeah, well, yes, Dr. Ledesma can recruit another honor student, or a new student, but we.
Do we have time for one more question?
I think
we had
more,
one.
and then I'm gonna ask Dr. Ledesma to come up and give you your
Okay,
report and
yeah.
so.
What other species are there of like something squirrels, foxes, and coyotes?
Yeah, we actually saw a lot. There were a lot of raccoons and skunks. And then, of course, like I said, squirrels. It was mostly mammals. There was an armadillo at one point. And then we had a camera in the Eastwoods that wasn't represented in this data set, but there was an otter actually that was captured. So we do have quite a lot of diversity. So besides looking at foxes, this is pretty revealing about what we have on campus, mostly capturing mammals as they're large enough and it captures the motion more easily.
That's awesome with the otter.
Yeah.
I'm curious about that. I get to now invite up Dr. Ledesma, who worked with Iman on this fascinating project. He'll get to say a few words about working with her on this thesis project, and also present the cord and stole, which Iman will wear at graduation, to represent this amazing work and also everything that she's achieved in the honors program. So congratulations and thank you.
So it's been really a privilege to work with Iman. This is my first year at St. Edward's as a professor, and I really lucked out working with such a great student. I had this crazy idea to study the foxes on campus, and Iman volunteered. She was really enthusiastic about working on the project, even though it involved potentially collecting fox poop. So she worked with me. I don't come from a mammal background, but Iman worked with me and we developed this project together really collaboratively, and Iman played a really big role in that. Beyond this research project, Iman has been a great leader on campus, president of the birding club on campus, leading a lot of birding walks, also helping to develop the birding class that Dr. Steve Fletcher teaches. And so Iman is a really great student, very proud to work with her, and I'm sure is going to go on to do great things. Iman, congratulations.